Okay, great. So I'm the next speaker, and my name is Michael Rogawski. I'm a professor of neurology and pharmacology at UC Davis, and there are my slides. This is my second opportunity to be here at this event. I thank Amber for that. A lot of my work has already been done by the queen of GABA and by Caddy. They're going to be a hard act to follow for me. But I'm going to try to talk in general about pharmacological principles that we could think about in ways of using the vast pharmacology of the GABA system to try to develop, I think somebody talked about temporizing treatments before we jump into definitive treatments like the gene therapy approaches that you'll be hearing about later. So I'm going to start with a tribute to this woman who some of you may know. Her name is Karen Gale. And I show her here for two reasons. Number one, she was a great neuropharmacologist and researcher in the epilepsy area. And I'm going to talk in just a few moments about some principles that she developed with respect to the role of GABA in the nervous system and in the functioning of seizures and epilepsy that are very relevant today. But I also wanted to highlight another thing about Karen Gale, and that is the fact that she transitioned from neuropharmacology research in general to the role of GABA in epilepsy when her daughter developed epilepsy. And so her commitment to this field is really remarkable, and it's emblematic of the commitment of so many other mothers who have devoted their energies to research in this field. And I think we can all celebrate the mothers who have made it possible for us to be here and to advance science in this area. And Karen was certainly an example of that. So what Karen said in her science is summarized on this slide. She emphasized, as you've heard already today many times, that GABA is the major inhibitory neurotransmitter in the central nervous system. And when we interfere with the GABA system, that causes overexcitation of neurons, and it ultimately can lead to seizures. However, Karen emphasized the fact that it's really not so simple. The dynamics of the GABA system are complicated, and depending upon which regions of the brain are involved and how the regions of the brain are affected, you can not only induce seizures, but you can have an inhibitory effect. You can not only have an inhibitory effect on seizures, but you can actually also induce seizures. And I'm going to give several examples of how excess activity in the GABA system can actually lead to seizures. And one of the important concepts that she promoted during her research career was the idea that the brain is not just simply a bag of neurons in a soup, but that there are specific regions in the brain that can control epileptic seizures in very specific ways. And I show on this slide an example of two areas that she focused on that were extremely important in regulating seizure activity. The first one on the left-hand side of the slide is called the area tempestus, which is in the deep piriform cortex here. You can see where that is located. It's a very discrete area. And also the substantia nigra pars reticulata is another critical area for seizure regulation. And if you enhance GABAergic activity, inhibitory activity in these brain regions, you can have a very profound anti-seizure effect. but not so much in other areas. And so the point here is that we may not need to modulate GABAergic function everywhere in the brain in a disorder that is associated with a mutation in a transporter that is everywhere. But if we target specific areas locally, we may be able to produce the desired effect. And so I just wanted to get people thinking about this concept that we may want to use the animal models that you heard about earlier to conduct experiments where we make focal changes, either with gene therapy approaches or other kinds of treatments that might be able to have a global impact on the brain. So how does this kind of thing occur? Well, these specific regions that are involved in regulation of seizure activity are hooked up with both inhibitory and excitatory neurons. So, for example, the superior colliculus Karen identified as a very important area in seizure regulation. When that area is excited with excitatory input, and glutamate is the major excitatory neurotransmitter in the central nervous system, it leads to seizures. The substantia nigra, as I mentioned, is a critical anti-seizure area, and it uses GABA to inhibit this superior colliculus area. But notice that these GABA neurons in the substantia nigra also receive a GABAergic input. So if GABA is enhanced, GABA activity is enhanced onto the substantia nigra neurons, that would inhibit them and cause a disinhibition here, leading to seizure activity. So you could see that enhanced GABA activity in this region can lead to a paradoxical excitatory effect. And so GABA is not necessarily only going to be inhibitory when its activity is enhanced, for example, with a mutation in the transporter that causes enhanced synaptic GABA function. Okay, so just very briefly then, I will run through some concepts about GABA as a neurotransmitter that you already heard from Amy Brooks-Kayal, and then talk in a bit of more detail about mechanisms by which alterations in GABA tone can actually lead to seizures. So this is a typical cortical circuit now, moving from the deep structures into the cortex. Basically, we have excitatory neurons, the so-called pyramidal neurons here in the cortex, and inhibitory interneurons. These are the GABA neurons. And these inhibitory neurons inhibit the function of the excitatory neurons, and we have essentially a balance. Sometimes we refer to that as a seesaw between excessive excitation and inhibition. You can get seizure activity both by excessive excitation and by having impaired inhibitory tone. You've seen schematics like this already a lot today. This is the synapse between the inhibitory interneurons and the excitatory neurons. GABA is packaged in synaptic vesicles in the inhibitory neuron. When the inhibitory neuron is excited, it releases GABA into the synaptic cleft. The GABA diffuses across the cleft, activates receptors on the postsynaptic membrane, and the GABA A receptors are particularly relevant. I'm also going to talk a bit about GABA B receptors if I have time. All of these receptors can get overactivated when the GAT1 transporter and the presynaptic nerve terminal, or in astrocytes, as you've already heard today, is defective. And that can lead to, in some cases, enhanced GABAergic activity, but in other cases we can get paradoxical effects, which I'll show you in just a moment. You've already heard about the structure of the GABA A receptor. We refer to the synaptic event that's produced by activation of the GABAergic interneuron onto the excitatory pyramidal or principal neuron as inhibitory postsynaptic potential. Usually this is hyperpolarizing. In some cases, it may just clamp the membrane to the reversal potential for chloride, and that tends to dampen the activity of the excitatory neuron. You've already heard today about the fact that there are multiple isoforms of the GABA A receptors. The synaptic isoforms that contain, for example, alpha 1, 2, or 3, and then the extrasynaptic ones that may contain alpha 4 in combination with delta. And you've also heard that the extrasynaptic GABA A receptors tend to mediate tonic inhibition or just set the resting potential of the neuron and are not involved in inhibitory postsynaptic potentials of the kind that occur with activation of these synaptic GABA A receptors. Now, there are many, many drugs that can affect the GABA system. We use them in clinical practice every day to treat anxiety, to treat seizures, to treat patients who require help going to sleep. Zolpidem is a very commonly used sleeping pill that acts on the GABA A receptors. So we have many drugs in the pharmacopoeia that can be used to modify the activity of the GABA system. At the moment, we don't know which, if any, of these drugs might be useful to treat a disorder where you have a defect in the GAT1 transporter. And I think there's an urgent need now to use these animal models that are just coming into the fore to try to determine what drugs could be useful, what drugs might be harmful in this situation. I mentioned at one of the question and answer periods that we do use clobazam in this disorder. That's a positive allosteric modulator of the GABA A receptors, the synaptic GABA A receptors. It should enhance the function of GABA. That seems paradoxical, right? If you have excessive GABA, why would that work? We need to understand that. That's a question. We have many drugs that can inhibit the function of GABA on GABA A receptors. I just list some of these here: pentyline tetrazole, picrotoxin, bicucline, a molecule called tetramine that we're working on in my research laboratory. Could very low amounts of these inhibitors be useful in patients with the GAT1 mutations? We don't know that. We would be certainly very cautious about using these drugs because we know that under normal conditions they can cause seizure activity, right? But maybe low doses of these kinds of agents might be helpful. The animal models give us an opportunity to evaluate that. And we have tiagabine, which you've already heard of today, which is kind of a pharmacological tool in a way that mimics the transporter defect. And I'm going to talk about what happens when you administer tiagabine to animals. This is an older paper from Randon Chapek's group at the Montreal Neurological Institute in Canada. What they looked at was the effect of tiagabine on synaptic activity in the hippocampus. And I think the results are quite intriguing, because when you give low-level stimulation of the GABAergic interneurons here, and this experiment was done under conditions where the excitatory function was inhibited by blocking AMPA receptors with CNQX and NMDA receptors with CPP. So you just have a situation where you're activating the inhibitory system, activating the inhibitory interneurons, and looking what happens to the excitability of the postsynaptic cell. And with low-level stimulation, you see you get inhibition here, and that inhibition is enhanced by tiagabian, which is the lighter curve. And with a higher intensity stimulation, you get a bigger effect, and that bigger effect is enhanced even more. And you'd expect that because what's happening is as you give higher stimulation intensities, more GABA is being released. That GABA is ordinarily inactivated by uptake into the presynaptic terminal because of the GAT1 transporter and also into astrocytes. If you block the uptake mechanism, then the GABA stays around longer and you see a larger signal that lasts longer. Now, if you give a very high frequency stimulation of the kind that might occur during an epileptic seizure, you see a profound effect of tiagabian to enhance the inhibition. But if you give even more stimulation, you can see what happens here, that you reverse the effect from inhibition to a very large excitation. And Amy touched on this very briefly when she said that the GABA receptor is usually inhibitory. But under some conditions, it can actually be excitatory, and she knows very well about that. She's a pediatric neurologist, and it turns out that very early in life, it's believed that the GABA receptor can be excitatory by virtue of the fact that the gradients for chloride have not reached their adult levels, where you have high concentration of chloride in the interior of the neuron. In the immature brain, that transporters for chloride are not well developed. And so what you have is a situation where the GABA receptor can actually be depolarizing and excitatory. And in the presence of a inhibition of the GAT1 transporter with this drug, and of course that's similar to what happens in the mutant situation, you can get a very strong depolarization, and you potentially could get generation of seizure activity. Now, I've only got a few more minutes, so I'm going to rush quickly through and point out that tiagabian, one of its toxicological effects is to produce seizures. This is paradoxical. Why would a drug that is normally used as an anti-seizure drug and can inhibit the transporter cause seizures? Well, at high doses, it does that, and I think you could see from the previous slide why that might occur. Here's a case of a patient with epilepsy who had his first case of very severe epileptic seizures called status epilepticus when he was treated with tiagabian, a paradoxical effect. And you heard that the mutant mouse model that was originally developed by Henry Lester at Caltech had a syndrome that was similar to the transporter mutation in humans. And what was not mentioned is the fact that these animals did have a reduced seizure threshold. Not a particularly profound effect, but there was a reduced seizure threshold in these animals. I think I'll skip that slide and jump to another concept that I wanted to communicate this afternoon, and that is the fact that while activation of the GABA system under many conditions, particularly in the adult brain, has an anti-seizure effect, in the immature brain, at very early times, you may not see much of an effect at all. But then, as the animal, or presumably as the child gets older, you can move into a situation where activation of the GABA system can, with activation in certain specific areas, lead to profound proconvulsant activity. And this is one example here where Mucimol, which is an agonist of the GABA A receptor. It basically acts like GABA, but it comes from a mushroom, and it can be administered systemically, and it gets into the brain, unlike GABA, which doesn't. And when mucimol is administered systemically, it causes an inhibition of seizure activity. But when it's injected into the substantia nigra here, you can see in the immature brain, it causes a profound reduction in seizure threshold. The latency to the activation of seizures is markedly shortened. So this is a situation where activation of the GABA system can lead to seizures. And this is work from Nico Moshe's group at Albert Einstein in the Bronx. And they used a variety of agonists, mucimol, and also an agonist called THIP, which is an agonist of extrasynaptic GABA A receptors. And what you can see is that in adult animals, normally these drugs at lower doses produce an anticonvulsant effect, but then as they get to higher doses in the adult, they can activate seizures. And in the immature brain, you only get this activation of seizures. So it turns out that GABA really has a double edge. It's a double-edged sword. In some conditions, it can be anticonvulsant. In other conditions, it can be proconvulsant. And finally, in just the minute or two that I have left, I wanted to touch on this idea of the GABA B receptor. And so there are, in addition to the GABA A system, which is the major inhibitory neurotransmitter system modulating excitation, you have this GABA B system, which is an entirely separate system that involves not what we refer to as ionotropic receptors, that are receptors that allow an ion to flux, in this case chloride for the GABA A receptor. But in the case of the GABA B system, these are what are called G protein coupled receptors. They don't have an ion channel that is intrinsic, but they do couple through second messengers to ion channels, including calcium channels and the inward rectifying channel called GERK. And the calcium channels are located presynaptically. GABA B receptors are presynaptic, and what happens is when GABA activates the GABA B receptors, it inhibits the calcium channels which are present on the nerve terminals that cause the release of GABA, and thus you have less GABA being released, and that would have a pro-excitatory effect because of the deficiency of GABA being released. And similarly, with respect to the postsynaptic effects of GABA B receptors, because they are postsynaptic as well as presynaptic, through a second messenger, it links to this potassium channel, which causes a hyperpolarization of the membrane. And I'll just end quickly with this slide, which somebody told me that they remembered this from my talk last year, so I wanted to mention it again just to reinforce this concept, which is that when the GABA B receptors are activated and activate this GERK potassium channel, it causes hyperpolarization of the neuronal membrane, and you'd think, well, that should be inhibitory, right? But it turns out that what you can do is deactivate a certain kind of calcium channel. Actually, Amy Brooks-Kale touched on this just very briefly. And when this calcium channel is turned on at these very hyperpolarized levels, and then the neuron repolarizes and becomes activated, it can lead to burst firing and epileptiform activity in the thalamocortical system, for example. And so this is a way that GABA paradoxically can lead to the generation of seizure activity. By causing this GABA B-mediated hyperpolarization, deactivating T-type calcium channels, it can lead to epileptic rhythmicity and spike and wave activity. And you, in fact, can see that if you inject baclofen, which is a agonist of the GABA B receptors, it's a drug that's used to treat spasticity. But in an experimental situation here, it can be administered directly into the thalamus with a microinjection. And when you do that, you activate seizure activity. So here's a substance, GABA, which ordinarily we think of as being inhibitory. It's acting on a receptor, the GABA B receptor, that we think of ordinarily as being an inhibitory receptor. And yet it's activating seizure activity. So we now have some understanding of why that might be, causing burst firing through deactivation of T-type calcium channels, and also, as I mentioned, blocking the release of GABA by inhibiting the presynaptic GABAergic nerve terminals. So with that, I'll stop and happy to have a discussion on any of these topics. Yeah, Katty.
Have you ever injected GABA B receptor antagonist into mouse brain to block seizure?
Yeah. So that's a great question. And there is quite a bit of work that's been done on GABA B antagonists. And they do have anti-seizure activity in models of absence epilepsy. And in fact, I'm a little bit surprised that the pharmaceutical industry hasn't pursued this. So you can have both effects. You can have both inhibition and activation with baclofen. Depending upon where you activate the GABA B receptors, you get different effects. It's a really good point. Anybody else? And by the way, Katty, thank you for mentioning Carson. Carson was an exceptional high school student that worked in my lab in Sacramento. And when I heard he was going to be going to Vanderbilt, we hooked him up with Katty, and he's having a wonderful time working with Katty. And thank you for mentioning him in your talk.
Carson is great, so a lot of behavioral writing reflex was done by him. He actually had made some comments, but I muted that. Yeah.
Thank you, yeah.